Here's a puzzle. Identical twins share 100% of their DNA. Every gene, every sequence, identical. And yet as twins age, they can become remarkably different. One develops type 2 diabetes, the other doesn't. If it's all in the genes, how can twins with identical genes have such different fates? The answer lies in epigenetics, a layer of biological information above the DNA sequence that determines which genes are read. It can be profoundly shaped by your environment, your experiences, and even the experiences of your parents. The word epigenetics literally means above genetics. It refers to changes in gene expression that do not involve changes to the DNA sequence. The DNA code is the same. What changes is how that code is read. Think of the genome as a piano. All 88 keys, all 20,000 and more genes, are present in every cell. But a Beethoven symphony doesn't use all the keys simultaneously. Epigenetics determines which keys are pressed. That's why a liver cell and a neuron, with identical DNA, perform completely different functions. Analogy. Epigenetics is like the volume knob, mute button, and equalizer on your genome. There are two primary molecular mechanisms of epigenetic regulation: DNA methylation and histone modification. DNA methylation is the addition of a methyl group to the cytosine base in the DNA, specifically at sites called CpG dinucleotides. This is performed by enzymes called DNA methyltransferases. When a gene's promoter region becomes heavily methylated, the gene is typically silenced. Methylation physically blocks transcription factor binding and makes the DNA inaccessible. Importantly, DNA methylation patterns are inherited when cells divide. DNMT1 recognizes the methylation pattern on the original strand and copies it to the new strand. Science note: abnormal DNA methylation is one of the hallmarks of cancer. In many cancers, tumor suppressor genes, which normally prevent uncontrolled division, become abnormally silenced. Cancer is as much an epigenetic disease as a genetic one. The second major epigenetic mechanism involves histones, the protein spools around which DNA is wound. DNA wraps around histone octamers to form nucleosomes, the basic units of chromatin packing. The tails of histone proteins can be chemically modified in dozens of ways. Acetylation, performed by HATs, weakens the grip on DNA. Chromatin relaxes, and genes become active. This is removed by HDACs, which we saw in video 4 are inhibited by butyrate from gut bacteria. Methylation by HMTs can either activate or repress genes depending on which histone is modified and where. Phosphorylation often marks sites of DNA damage or chromosomal condensation during cell division. Together, this pattern constitutes the histone code, a language that cells interpret to express or silence genes. Environmental exposures can modify epigenetic marks, altering gene expression for years or even decades. Adverse childhood experiences have been shown to alter DNA methylation in stress response genes like NR3C1. These changes affect how the stress response system is calibrated for the rest of a person's life. The Dutch hunger winter of 1944-1945 provided a tragic natural experiment. Children born to mothers pregnant during the famine showed altered DNA methylation at insulin genes decades later. Cigarette smoking leaves distinctive DNA methylation signatures, some of which persist for over 30 years after quitting. Certain industrial chemicals and air pollutants similarly alter epigenetic marks, contributing to cancer risk. Perhaps the most startling frontier: can epigenetic marks be transmitted to subsequent generations? Male mice exposed to fear conditioning show stress responses in their offspring, transmitted via marks in sperm. Grandsons of men who experience famine during prepuberty show altered mortality patterns. Most marks are erased and reset during the formation of eggs and sperm, a process called epigenetic reprogramming. But some marks clearly escape this erasure, and the implications for how we think about heredity are profound. Epigenetic mechanisms are now major drug targets. HDAC inhibitors are approved cancer treatments. DNMT inhibitors reactivate silenced genes in blood cancers. The epigenome is now a programmable layer of biology. The epigenome, once thought to be the static backdrop to genetics, is now recognized as dynamic and druggable. Our genes may provide the script, but epigenetics is the director. It is the bridge between our environment and our biology. Visit younbedsidemanners.com for more stories from this series and others. Thank you for exploring the frontiers of medicine with us.